Hello again. I'm Steve Piantadosi. I'm going to moderate the final session in the workshop. This is session 6, the challenges and successes of review and oversight of multi-center cancer studies. We've divided this session into two principal themes. The first being the perspectives of the PI, trial sponsor, and host institution. We have two experts to discuss this perspective. Barbara Beer, who's the Senior VP of Research at Brigham and Women's, and Rich Schilsky, who's the Chief Medical Officer at ASCO. The second perspective is that of local and central oversight bodies, and that perspective will be discussed by David Parda, who's the System Chair of the IRB at Allegheny Health Network, and Chris Dougherty, who's an IRB Chair at University of Chicago. So we'll take our discussants in that order, beginning first with Barbara. Oh, Rich, you guys swapped. Okay, I'm sorry. Rich, you're up. Thank you.
Thanks, Steve. Good morning, everyone. Good to see so many people still hanging around at this point in the meeting. Just to start off, for those who don't know me, let me just tell you the perspective I'm coming from. I've had about a 30-year career as an academic clinical researcher, nearly all of that at the University of Chicago until about a year ago when I joined ASCO as the Chief Medical Officer. I did serve for 8 years as Associate Dean for Clinical Research during my time at the U of C. I also spent about a decade as the Director of the Comprehensive Cancer Center while I was there. And for 15 years, I was the group chair of the Cancer Leukemia Group B, one of the cooperative groups that is now merged into the Alliance under Monica Bertagnoli's leadership. And obviously, the challenges relating to multi-site studies and IRB review depend, of course, on your perspective. And I've just listed here many, if not all, of the perspectives that come into play. For purposes of this session, I'm going to sort of portray the perspective of the research sponsor or the PI on a clinical study. So, of course, the issues from that perspective, I think, boil down to things that almost everyone here is well familiar with. First is the efficiency of study launch. I can tell you that when I was running the CLGB, when we had about 250 sites, and we would activate a protocol, meaning that the protocol was ready to go to all the local IRBs for review and then to begin accrual, it would take at least 3 to 6 months from the time of activation until the first patient was enrolled in the study. It would take typically a year before the rate of accrual began to approach what the expected rate of accrual for the study was, simply because of the time to get IRB approval at all of those participating sites. This is particularly a problem if you're doing some sort of a time-sensitive study. You know, if you're trying to study the causal effect, the cause of an epidemic, for example, that may peak and then wane in a short period of time. If it's going to take you longer than the period of time for the epidemic to get IRB approval to actually study its causes, you're going to miss the opportunity. From the perspective of the sponsor, of course, there's the whole issue of redundancy of review when there are a few changes permitted in the protocol. Again, I can tell you that, at least from the perspective of a cooperative group, we routinely would get queries from sites about, some aspect of the protocol or another. Rarely, but occasionally, were they, substantive scientific commentary that would actually lead us to rethink the design of the protocol. Most of the time, they were not. And, our option was to tell the site, if you, don't feel you can participate in the protocol, don't, because we're not changing it. There are issues, of course, surrounding variable quality and extent of local IRB review that potentially would diminish information transfer to study participants, or even potentially increase the risk to study participants, depending upon what the local IRB, how the local IRB modifies the model consent form for the study. It was often the case, of course, that there was an increase in the length or complexity of the consent form following the local review. I mean, every protocol that we sent out from the cooperative group had an NCI-approved model-informed consent document that was written by the investigators, approved by the NCI. In those years, mostly not approved by the NCI central IRB, but just approved by the NCI staff. So there have been several layers of vetting of the model consent form, which is almost never then just adopted wholesale, of course, by the local institution. And sometimes we would find on auditing that certain required elements of the consent form were dropped out by the local IRB for some reason or another, which gave us considerable concern about the quality of information that was being transmitted to the research participants. There are not now, I don't believe, any standard metrics to define what is a high-quality IRB review, so it's really difficult to know, looking across multiple sites, whether an IRB is doing a good job. And then, as I'll come to at the end of my remarks, of course, I believe that there is and will have to be a changing recruitment model for the biomarker-driven clinical trials that are increasingly common in oncology research these days. There's very little empirical research about the impact of central IRBs. There was a very nice review article that was published at the end of last year by Czech and colleagues, and I've just summarized on this slide and the next a few of the key observations that were recorded in that review article, but I would urge people who are interested to actually look at the paper. So after an extensive literature review, they could only identify 11 empirical studies on IRB review of multi-center trials. And some examples of things that they found in these 11 studies, a study by Ravinia et al., there were an average of 5.2 changes made per site in the protocol or consent form, but generally in the consent form there were no substantive changes made to the protocol. The estimated cost of the local IRB review process, I think this was for a 25-site study, was over $100,000 devoted to the local IRB reviews. In a study by Stark and colleagues, of 18 IRBs, 16 of the 18 IRBs in a multi-center study had changes to the informed consent form. 7 of the 18 IRBs had no concerns with the protocol, but 9 had at least one major concern. In a study by Berman et al. Of 25 sites participating in a multi-center study, no changes were made to the protocol as a result of the local review, but there were a median of 46.5 changes made to the informed consent forms by these 25 sites, of which 82% changed the wording without changing the meaning of the informed consent document, and the median time to approval across the sites was 104.5 days. Silverman and colleagues did a study of 16 sites participating in a multi-center trial. Only 3 of the 16 informed consent forms contained all of the required elements. This gets back to the comment I made previously. And the reading levels range from grades 8 to 13. We had some discussion about this yesterday. So again, it's not clear that the local IRB review is actually improving the transmission of information to the research participants. From all of the studies that were cited in this review article, the conclusion in general was that local IRB review of multi-center studies was, of course, highly variable. Changes made to the protocol were rare. Changes to the informed consent form were common, and there was a risk that they could introduce misinformation and that much time and effort was consumed by all parties. And then there was a recent review article by Wagner et al. Summarizing the NCI central IRB experience, showing that, compared to, I guess, historical data from the NCI CIRB was associated with faster reviews, less staff effort, and reduced cost. I can tell you that I was around, as was Michelle Christian, when the NCI Central IRB was just implemented, and it was painful in the beginning. There was no question about it. The reviews took an exceedingly long period of time. They were of highly variable quality. They were not transmitted in any standardized format. It was impossible to know, what the Central IRB viewed as being really important versus what they considered to be trivial. And it was made it very difficult for us, who were essentially the sponsors of the research, to really figure out where the concerns were. And of course, there was not a lot of early adoption among the sites of the Central IRB process. As far as I know, a lot has changed in the 15 years or so since the NCI Central IRB was introduced. The reviews are much more timely. They're much more relevant. They're well delivered, and there is increasing and fairly widespread adoption. So all for the good. Now, I don't know if Dr. Menikoff is still here, but he, of course, has written about his views on multiple IRB review in a paper that he published in the New England Journal a few years ago. I've summarized his views as expressed in that commentary piece about problems with multiple IRB reviews. They diffuse responsibility, potentially leading to more superficial review, because no one IRB feels like they have the true responsibility for the research. No IRB feels empowered to change the protocol. If there are substantive problems with the protocol that are identified by an IRB, they may not be communicated because the site just chooses not to participate in the study instead of raising their concerns with the study sponsor. The consent forms that accompany most multi-center studies in most cases are already compliant with regulatory and ethical standards, and local changes can introduce errors or confusion. And again, substantive problems noted by a local IRB with the consent form might not be communicated broadly to all the sites participating in the study. So those are all things that I think we have to think carefully about. So just again, looking at some of the challenges for the various stakeholder groups, from the perspective of the sponsor and the principal investigator of the study, there are major issues regarding efficiency of study startup. If you're not using a central IRB, there's substantial workload required by the study sponsor to meet the needs of all the sites. You put a protocol and a consent form out there to 200, 300, 800 sites, you get a lot of questions. And if you're the study sponsor, you have an obligation to respond to all those questions. And then you need to track, to some extent, what the variation is across all the consent forms, because the consent form ultimately determines how you can conduct the study at that site. And where it becomes particularly important is regarding consent forms for some of the correlative or ancillary studies or biospecimen banking. You have to be able to know exactly what each participant in the study consented to with respect to the use of each biospecimen that has been collected. And no biospecimen can actually be distributed for any research purpose unless you're sure that it's been appropriately consented for that specific research purpose. And to the extent that those modifications can be made locally across different sites, it becomes incumbent on the sponsor to have mechanisms in place to track all those things. From the perspective of the local IRB, I mean, that's not really the position I'm addressing right now, but just some thoughts. Of course, there's the time, effort, and cost of review, the obligation to protect local participants in the study, concerns about institutional liability if you sort of give up your local responsibilities to a central IRB or another IRB of record. There's this temptation to make changes because, that's what you give a group of people a job. They're going to feel obligated to justify their time and effort by doing something, and they tend to make lots of changes. And then, of course, there's an issue about lack of awareness of the experiences or concerns raised at other sites participating in the study, which are often not communicated among all the different sites that are considering participation in a protocol. For the research participants, I think there are many issues as well. How can they know if they've had a good quality IRB review of the study that they're being asked to participate in? How can they know if they've gotten complete, accurate, and understandable information about the risks and benefits of the study? All they know is that there's a doctor sitting there in front of them asking them to sign a piece of paper after some level of discussion, and there's no way for them to assess whether or not the information that they're getting is better or worse or different from information that other study participants might be getting at other sites for the same protocol. And then, of course, might they miss an opportunity to participate in the study if their particular hospital or clinical site declines to participate in the protocol? And if it's a study that they feel that might really benefit them, do they then have to seek care elsewhere to get access to that same research study? So just to conclude with a few remarks about IRB review in the era of genomic medicine. So, of course, life is getting a lot more complicated because increasingly we have a need to identify patients with rare genomic subtypes. It is, in my view, increasingly impractical to seek local IRB review at multiple sites for occasional patients. And then I think we have to ask a legitimate question about whether every local IRB has the expertise necessary to review these increasingly complex biomarker-driven studies. So here's the problem, of course. Every cancer that we deal with increasingly is becoming a group of rare cancers. So, when I was starting out, there was non-small cell lung cancer and there was small cell lung cancer, and that was about it. Then we realized that there were differences between adenocarcinoma and squamous carcinoma in the non-small cell category. But now, if you look at adenocarcinoma of the lung and squamous carcinoma of the lung, we are quickly approaching dozens of small molecular subtypes, each of which may have a different presentation, a different associated phenotype, a different prognosis, and may require a different treatment. And if we're going to start studying these as unique diseases, lung cancer as a common disease is increasingly going to become a bunch of rare diseases. And the same, of course, is true for virtually every other kind of cancer. So if you're going to try to do a clinical trial where you match a drug treatment to a particular genotype in the tumor, if I want to study a MET inhibitor or a BRAF inhibitor or a PI3 kinase inhibitor in patients who have adenocarcinoma of the lung, I might have to screen 10,000 or more patients to get a sufficient number of patients to actually participate in the clinical trial that I want to do. And because all of these are relatively rare, I might have to activate my protocol at 1,000 sites to screen the 10,000 patients for the 1 or 2 patients at each site who might actually be eligible to participate. This is just simply not feasible. It's not economically feasible. I don't think it's feasible under the current regulatory framework, and it's I think, fraught with peril. Can we do anything differently? Yeah, we can enroll patients in so-called screening protocols. The advantage of that is you have one protocol. You enroll all the patients, say, with adenocarcinoma of the lung, and what you're asking their consent for is to do the molecular screening and then to follow their outcomes, but not do any intervention. This is really an observational protocol. It gives you the opportunity to do the screening, capture the genotype, learn about whether there's a genotype-phenotype correlation, learn about whether a genotype is associated with a particular prognosis or not, but doesn't require any intervention. It does give you a pool of profiled patients, if you will, who then could be offered participation in specific intervention trials. Or alternatively, that all of the genotyping and so on could be done outside of the clinical trial context, and only the patients who have the particular biomarker of interest are offered enrollment in the therapeutic trial. That is the hit or miss approach in a sense of, if you happen to have a patient in your population who happens to have the relevant eligibility criteria for a rare genotype. So, historically, the model by which we've done intervention trials, at least, is the patient comes to the trial. That is, I have a trial portfolio at my site. I have patients who come to my clinic. If I'm lucky, if the patient's lucky, maybe I have a trial that's appropriate for that patient at that time. I think that's the model that it probably needs to change. We need to figure out how to deliver the trial to the patient, not the patient to the trial, because these patients are going to be, rare as hen's teeth. They're going to be scattered all over the globe. And what we have to be able to do is to get the trial with an associated IRB approval to wherever the patient is. I think that approach is going to require more central IRBs. As I said, individual IRB review just becomes impractical and excessively costly for the number of trial participants who might be expected at each site. So then just to conclude, it seems to me increasingly that little is gained from individual site review in multi-center studies. There is potential for multi-site review to actually increase risk and diminish the quality of information conveyed to research participants. Recruitment in the genomic era is, I think, really only practical with the use of central IRBs. We need a regulatory framework that will encourage site acceptance of central IRB review. And of course, we need some additional research on the appropriate metrics to define what is a high-quality IRB review. So I will stop there, turn it over to Barbara.